So this can be quite different from the last talk. The purpose of this project, and the sort of vague title doesn't expressly explain it, was to really take a step back and think about how we should go about sequencing and generally molecularly characterizing tumors with next-gen sequence technology. And the reason that we're really sort of stepping back and thinking about this from first principles a bit is because of this rather obvious observation that tumors are very impure often, and they're often heterogeneous, and they're often aneuploid. And each of those things really complicates sequencing. So you have this problem that your tumors often contain normal cells, and that reduces the signal that you're really looking for, which is mutation. And similarly, tumors are often a collection of genetically diverse cells with different origins, with complex relationships, and that can also influence your ability to really characterize what the tumor is made up of and what the important mutations are. And similarly, a lot of analysis is sort of proceeding from the assumption that you have an approximately diploid genome, and this is, of course, not true in many, many tumors. So what we're really comparing to here is a body of work that has been done over the last 5+ years, which has followed a bit of a kind of a common recipe of either whole genome sequencing or exome sequencing. In the case of whole genome sequencing, it's been very typical to target 30x coverage of the tumor, or maybe 40 to 50 or 60x in some cases. And really, this is insufficient depth for sensitive detection of mutations, and it's particularly true if any of those things that I discussed on the last slide are true. So if the tumor is impure, if it's heterogeneous, if it's aneuploid, this amount of WGS data becomes increasingly inappropriate. So sort of the main alternative to sequencing tumors is exome sequencing, and there we typically target somewhere in the region of 100x average coverage. And this is certainly better, but then you introduce a new problem that you really have insufficient breadth to resolve things like clonal architecture. You don't have the coverage where you need to detect regulatory variants. Exome data is notoriously bad at detecting structural variation and copy number assessment. And then sort of on the RNA side, transcriptome analysis is to sort of follow this rote pattern of doing a lane of Illumina sequencing generally at most, but often considerably less than that. Generally, people are pooling 3 or 4 or 5 samples in each lane. So really, in a clinical context, these levels are really insufficient, and that's the argument that I'm going to make. And in the clinical context, you really need sensitivity and accuracy to be much higher. So in a research discovery perspective, you're improving sensitivity by sampling across many, many patients. You have 100 patients in your cohort. It increases the chance that you will catch everything important that you're looking for. If you just have one patient and you really want to understand their tumor, you're placing a lot of demands on the data. So to go through this exercise, we really selected one interesting case that we had previously sequenced, and it was an AML. And this data, or preliminary data from this case, has been presented in several publications. And this case was chosen because it appeared to be, at first glance, possibly interesting in terms of heterogeneity and clonal architecture. And the result I'm showing here is from the original sort of standard sequencing approach. So we did 30x whole genome sequencing, and we discovered this tumor which it went on to relapse. So this scatter plot is showing tumor variant allele frequency on the x-axis and the variant allele frequency from the subsequent relapse of that tumor on the y-axis, and showing sort of the things in the middle there that are mutations that are from the founding clone that are present in all of the cells of both the primary tumor and the relapse. And then there's some evidence that there were perhaps clones in the primary that were lost during evolution of the relapse. And also there are things that were really gained or enriched in the relapse. So we don't know for sure whether they were there in the primary, but we do know that they became much more prevalent in the relapse during treatment and relapse. So by analysis of this sort of preliminary data, we came up with an initial model of what we thought the evolution of this tumor looked like, and it's depicted here as a sort of fisheye plot, where in the primary we have a dominant clone and a subclone depicted in green. And both of those cell populations contain DNMT3A, FLT3, and IDH1 mutations, which are thought to be either initiating events or very close to initiating, and all subsequent cells that evolve from that tumor contain those mutations. And what was interesting was that in this particular patient, they went through chemotherapy, they were treated very successfully, they achieved a morphological remission, and then about a year and a half later, they had a rather drastic relapse. And the tumor that we sequenced at that time, it still had, we could tell that it was clearly from the original founder tumor, but it now was enriched for a subclone that was actually very, very rare in the primary tumor cell population. So to really go back and say, well, what if we sequence this tumor, the primary and the relapse, as if money was no object, to try to get a sense of what the optimal sequencing strategy might look like. So we went back and sequenced the whole genome to about 350x and sort of tackled various factors of a sequencing experiment by sort of expanding and trying experiments at every stage. So we did multiple independent libraries. We selected different sizes of DNA fragments for those libraries and produced replicates of each of those. We did about 350x exome sequencing using 6 different exome reagents or providers. We selected 200,000 putative single nucleotide variant sites and designed a custom capture reagent, and then used that to capture DNA and sequenced that to about 1,000x. We also did additional targeted sequencing of a panel of genes that are found to be recurrently mutated in AML and sequenced that to about 1,200x. And then a further subset of these sites were sequenced to 10,000x, and then a further subset was profiled by digital droplet PCR to almost 250,000x. And then instead of the sort of standard 1 to 1/4 of a lane of RNA-seq, we produced 30 lanes of RNA-seq data, which works out to about 6 to 10 billion paired 200 base pair reads for a single library or a single sample. So this deep characterization, of course, we produced a huge amount of data, which is just going to increase to the challenges that were discussed in the last presentation. And of course, this led to a lot of analysis opportunities. And we also expanded the analysis throughout time, so sampling multiple time points throughout the evolution of this patient's tumor over the sort of several years that they've been tracked in the clinic. And the analysis really led to a number of outputs. So we did things like stepping back and saying, are our alignments any good? Should we try different alignment algorithms? We did a pretty comprehensive comparison of variant callers, tried to really look at regulatory analysis by integrating the DNA and the RNA data. And I don't have time to go into many vignettes from all of those things. But one of the principal outputs of all of this analysis was to develop what we felt was a really high confidence platinum list of somatic variants. So a subset of these 200,000, which actually were a subset of 400,000 predicted somatic variants that we feel are actually very high quality and likely to actually be real and across a wide range of VAFs. So not just the things that are easily detected from the primary clone, but things that are really confident but from very rare subclones. And then, of course, having all this data allows us to do sort of retrospective downsampling experiments. So we went through this discovery and validation phase and identified variants that we felt were very high confidence, and then we went back and we downsampled the whole genome data to various levels, and we looked at the effect of that downsampling on sensitivity to detect variants. And what's shown here is the result of 10 iterations of that downsampling experiment to 2 different target depths. So the top one is 30x and the bottom one is 50x. And then the color coding indicates whether you found the sort of true positive variant again. How many of those 10 attempts were you able to find the true positive variant? And if something's red, that means that it was never found, even when we randomly sample 10 times to 30x or 10 times to 50x. And basically what we found is that anything below 20%, you have a very low chance of actually being able to detect those things. And even some of the variants that are at much higher variant allele frequency, again, you really are not able to detect them at the 30x to 50x threshold. That is still very common in experimental designs for this, for tumor profiling. So similarly, we looked at multiple variant callers. So we evaluated initially 11 or 12 variant callers. About 7 or 8 of those sort of made it through the preliminary evaluation. And we looked at different ways to improve sensitivity and accuracy by perhaps combining them, and we found that taking an intersection of some subsets of these colors gave you a pretty decent balance between sensitivity and positive predictive value. And if you want to talk more about the details of some of those variant colors, you can come and find me afterwards. In terms of the biology of this tumor, one of the things we found, which I guess in retrospect wasn't that surprising, is that as we sequenced deeper and deeper, we found more and more interesting things. So we didn't just find more somatic variants that are perhaps meaningless. We found somatic variants in important cancer genes and at important positions in those genes. So I'm showing here a list of some of the somatic variants that are sort of deemed to be really relevant to AML biology. Only the top 4 were found in the initial whole genome sort of standard sequencing approach, and the remainder were found by the deep sequencing approach, and these include canonical mutations in FLT3 and RUNX1 and IDH2. So not just any mutation in these genes, but actually well-established driver mutations in those genes that were completely missed with the sort of standard sequencing approach. And when we look at the sort of the clonal identity of these mutations, we see that they fall into several categories or several putative subclones. So we have NPM1 and DNMT3A being in the founding clone, and these things are carried through from the primary tumor to the relapsed tumor that evolved from the primary. But then we have, in each of the putative subclones, we also have putative drivers of those subclones. And some of these subclones along the bottom of the x-axis are in clones that were actually lost during treatment and didn't come back in the relapse. So these are things that presumably responded the most strongly to the treatment. And then there are other things that really evolved out of the primary and became dominant in the relapse, including the dominant clone, which now had an IDH2 mutation. So if we look at sort of the overall distribution of variant allele frequencies in the primary and the relapse, that we can see that these seem to line up with very nice clusters of mutations that correspond to our subclone predictions. 6 of these clones are shown here, and then the panels around the main panel, you can see sort of the shift in that population of mutations from primary to relapse. So you have things that are really being gained or enriched as you go from primary to relapse, and you have other things that are really being lost as you go from primary to relapse. And again, if we do a downsampling experiment of some of our data, so we have a data set for all of these platinum variants that were sequenced to up to about 1500x in the primary and 300x in the relapse. We can see that when we start at the sort of 30x level and even maybe at the 50x level, we can see our initial 3 subclones. As we get up to 70 or 80x, we can start to identify a 4th clone, one of the ones that seems to be relapse specific. And as we go up to 200x, that resolves into 2 subclones in the relapse. And as we go up to closer to 800 or 900x, we start to see the 3 subclones that were each lost from the primary. And then it kind of stabilizes as you go beyond that. There's a link there. When you get a copy of these slides, you can go and look at a video that shows you sort of the smooth transition across increasing coverage in our ability to really resolve these subclones. So we started with this original clonal evolution model, which was quite simplistic. And we added on all this additional information, which allowed us to give a much richer picture where we have a larger number of clones, each with their identified driver mutations, and showing what the transition of these subclones from primary to relapse. And then we used the additional data that I mentioned from the various time points to actually show that that model is actually still incomplete, and there's what we thought was a subclone of the original tumor that has a p53 mutation that actually seems to be completely independent of the founding mutation. So the p53 mutation doesn't track over time with the DNMT3A and NPM1 mutations, and it appears that what is happening is actually there's another initiating cell that had a p53 mutation that during treatment sort of fills the void. So you have this p53 containing cell that becomes sort of an independent clone as you go through treatment and the tumor disappears. And then when the tumor comes back in relapse, this independent clone kind of shrinks back. So these guys are kind of in competition with each other. And this patient was eventually cured. We hope that they will stay that way. And they still to this day have a subclone population in their blood cells that harbors this p53 mutation of unknown clinical significance. So the sort of predictable take-home message of all of this is that with this sort of standard analysis and sequencing approach, you get a nice picture, but if you start to work on optimizing that and making it much more comprehensive, you get a much richer picture that sort of crystallizes out of the data. And of course, this can all be, documented and made into a formal protocol. And of course, this procedure that we described here is extremely expensive and complex and time-consuming and involves a huge amount of data. And we're not proposing that everyone should do this on every tumor, but we think that by making this data available and all of the analysis that went along with it, it'll help people sort of choose an optimal balance between cost and comprehensiveness in their own tumor sequencing projects. So I'll just end with acknowledging especially Chris Miller. So he and I really divided and conquered on a lot of the analysis. And then, of course, Tim, Elaine Martis, and Rick Wilson for their support at the Genome Institute. And I'll take any questions. Thank you. All right, thank you.